And here we go. We've talked about parasitic Chelicerata. The only real group of those is the Acari, the mites. We've talked about something just went ding, one moment. We've talked about Acari. We've talked about various parasitic crustaceans, most of which you are, here we go, most of which you're not that likely to find unless you go into pretty exotic veterinary practice, although there are a few that can parasitize humans, like those pentastomids, the so-called tongue worms. So I've said my piece about them. From here on to the end, everybody we're going to talk about is insects. Insects belong to a larger group called the Hexapoda. As you might expect, this means 6 feet or 6 legs in Greek. And hexapods have 3 pairs of walking legs, 6 in all, 3 times 2 being 6. Duh. There's only 1 pair of antennae. You might remember that chelicerates don't have any. They do have pedipalps, but that's different. Those are posterior to the mouth, whereas antennae are in front of it. Crustaceans have 2 pairs of antennae. Hexapods only have 1. Hexapods also have 3 pairs of mouth parts. Those are very highly modified legs, although in most species they don't look very leg-like. There's a 3-part tagmosis scheme, a division of the body into a head, a thorax, and an abdomen. The head's got the mouth parts and the antennae. The thorax has the walking legs. And the appendages on the abdomen are highly reduced or very often missing completely. The genitalia are modified appendages. Often in insects, those are tucked in and not visible unless you dissect them out. Now, this gives me a chance to tell one of my traditional humorous references. Identifying insect species is often very important because insects, aside from being parasites, include a lot of agricultural pests and things like that. Many insects can only be told apart by careful study of the genitalia. And so entomologists sometimes have to dissect out a lot of insect genitals, which is not nearly as exciting as it sounds. But some entomologists in Australia invented a very small gizmo that would inject an extremely fine, low-velocity stream of alcohol into insect genitals and cause them to turn inside out and extend out of the abdomen so that they could be seen and the insect could be identified. They called it the phalloblaster, which I just think is a really cool name. Anyway, genitals are modified appendages. They're not visible from the outside. Sometimes there's 1 pair of appendages on the very last segments that will often look like a pair of bristles. They're called the cerci. And yes, even though they're not spelled the same, it's always struck me as peculiarly important that things growing out of an insect's butt would have a name that sounds like the name of a town not too far from here. Every time I've driven through that town, it tends to confirm this impression. Apologies if anybody's from there. Okay, right. Non-insect hexapods include groups like the Collembola, aka the springtails. This is just one of them. It's a British one. None of these are parasitic as far as I know. This is just a reminder that not every hexapod is an insect. Collembola are extremely abundant in leaf litter. They're swarming in the decaying autumn leaves even as we speak, and they may have a role to play in breaking those down. You find them in soils. You find a lot of them on moss. They're very small, and they've got some specialized appendages on the abdomen that enable them to leap, hence the name spring tail. They can jump like they've got a spring in their tail. but they're generally less than a millimeter in size. You're not likely to see them unless you go deliberately looking for them. So there's a few hexapods that aren't insects, but insects are the great majority of hexapods, and in fact, they're the great majority of species on the planet. About 3/4 of all known species, about 1 million known. This includes 350,000 species of beetles and 170,000 known species of moths and butterflies and so on. And we keep discovering more. Of the undiscovered species that are left to discover, probably the great majority of those are insects. It's basically their world and we just live in it. There's an old story about the evolutionary biologist, British gent by the name of J.B.S. Haldane, working about 100 years ago. The story is that he gave a talk on evolution, and a sweet little old lady came up afterwards and says, you know, what have your studies of nature taught you about God? And Haldane, who was an atheist, said, he has an inordinate fondness for beetles. Right, like I said, it's their world. We're just living in it. What makes an insect, aside from being a hexapod, is the mouth parts are on the outside of the mouth. This is said to be ectognathous. Springtails and things are entognathous because the mouth parts are tucked in on the inside of the mouth. Insects always have 11 abdominal segments, although not all of them are visible from the outside because some of them are kind of turned inwards. The segments that bear the genitals may be kind of tucked in like that. But if you trace the way that they develop, there's always 11. And aside from parasites, they can be disease vectors. We've already seen this talking about malaria and sleeping sickness. In some areas, up to 1/4 of a crop may be lost to agricultural pests. On a more positive note, at least from a human perspective, they're important pollinators of a great many plant species, including an awful lot of crops. And of course, they produce honey, wax, silk, things like that. They're even eaten in some areas. I've yet to go to the part of Mexico where you can try chapulines, roasted grasshoppers, but one of these days I'd like to try those. So you've seen this slide before. We've talked about acari, we've talked about the parasitic crustacea. Centipedes and millipedes don't have any parasitic species in them. The parasitic insects include Anapleura, which is the lice. Actually, that's a slight typo. That should be the Pithyraptora. We can now reclassify those a bit. The Siphonaptera is fleas. Strepsiptera is this really weird group that I'm going to lecture on in some detail, mostly because they're creepy. Diptera includes flies, and of course there's a lot of micropredators among the Diptera, you know, horseflies and mosquitoes and biting midges and black flies, for example. But there's also some flies that are more permanent parasites. Estridae includes botflies, where the larvae actually burrow under the skin and mature there. Hippoboscidae, common name for which is keds, do much the same thing, especially on sheep. And then Hymenoptera is your wasp, bees, and ants. And one of the most diverse groups of all insects is the parasitoid wasps, which we will talk about in some detail, noteworthy for having been the inspiration for the Alien series of movies. There are a few other groups that may have parasitic larvae, but the adults may be free-living. We might mention those in passing, but I want to move ahead to the Pithyraptora, which is what we'll focus on today. That's the lice. There's that word, PHTH, Thyraptera. They're wingless and they suck blood. And we used to divide these into the Anopleura, which is the sucking lice. These are the ones that sometimes parasitize humans. And Malophaga, which is Greek for the wool eaters. Malophaga are chewing lice. We now know, however, that the Malophaga is paraphyletic. The last common ancestor of all Malophaga was also ancestral to the Anopleura, so we no longer divide the Thyraptera up in this way. So the sucking lice have piercing mouth parts, and they suck blood, and they suck in general. Chewing lice include a lot of species that parasitize birds. They'll feed on feathers, they'll eat dead skin. Some of them can eat living tissues and may drink blood as well, although they've got chewing mouth parts rather than stabbing mouth parts. There are some that can parasitize mammals. I can't think of any that are long-term parasites of humans, although I've heard of humans getting bitten by chewing lice if they come into contact with a lot of birds, like people who are plucking chickens, for example, might get chewing lice bites, but they're certainly not long-term human parasites. And here's the difference. On the right, that is good old Pediculus corporus. That's the human head louse. It might be I think it's a head louse. It could be a human body louse. It's one of the two. That's the one on the right. That's the one that can cause problems in places like elementary school. And you can see there is a join. There's a joint between the head and the thorax. The thorax is the part where the legs are coming off. The head is up at the front with the antennae, and there's a joint between them, but the head is more narrow than the thorax. On the left, that's a chewing louse, and the head is wider than the first segment of the thorax, the so-called prothorax. And this makes chewing lice look like they've got a big head with a prominent neck. You can see that the overall outline of the body is quite different. I'm assuming that's related to the functional need for more muscles in your head if you're chewing, as opposed to if you're sucking. I don't know, but that sounds reasonable. So that's the way you can tell the two groups apart. Oddly enough, sucking lice are not all that diverse. There's only about 500 species known. They only live on mammals, and in fact, they only live on 20% of mammal species. There are no sucking lice on elephants. There are no sucking lice on monotremes. That's the platypus and the echidna. There are no sucking lice on manatees. Okay, that's not a sentence you say very often. But yes, manatees are blissfully free of sucking lice. Okay, here we go. All right, had to admit somebody there. There are certainly human sucking lice. Humans get head and body lice, which seem to be in the process of splitting into two species, although they're not there yet. We also, of course, get pubic lice. Other animals get them too. That's a cattle parasite right there, Solenopotes. And one thing that was really cool when I found this out is that you might not think that marine mammals would be prone to getting sucking lice, because not only does a sucking louse have to hang on very tightly to its host, but marine mammals are constantly diving. And so any parasitic insect that has to make a living on a marine mammal has to cope with being immersed in cold water and diving possibly 1,000 feet down on a regular basis. So you wouldn't think that this would be hospitable, and yet there are a number of species of sucking lice that parasitize seals and sea lions. And this happens to be one of them, Echinipthyris horridus, one of the small numbers of marine insects. You know, if you think about it, insects have radiated amazingly on land, but very few have ever returned to a marine habitat. I'm guessing that's because the crustaceans are taking up all of the ecospace. But yeah, this is one of the few. This is a parasite on seals, which is weird. We can divide up those, what we used to call the Malophaga, the chewing lice, into Amblycera and Ischnocera. The Ischnocera, that's second from the left, they feed mostly on feathers, and they're noted for being extremely host-specific. A species of Ischnoceran may be restricted not only to one species of bird, but to one specific body part on one species of bird. Amblycerans are a bit less restricted, and there's a few members of both taxa that feed on mammals. I don't know much about them, and the vets that I have talked to don't see them around here, or at least not very often. But there are both dog and cat lice, and these are apparently in the Ischnocera. They're not related to human body lice in the Anopleura. So there you've got Amblycerans and Ischnocerans. There's a typical Anopleuran that's third from the left. And on the right, I think there's only a couple of species of Rhynchoptherina. They're restricted to living on elephants and warthogs. And you'll note that that Rhynchoptherinan has very long mouth parts. Given that elephant and warthog hide is extremely thick, you need mouth parts that are built like an oil drill if you're going to suck blood from an elephant. And this kind of thing is what Rhynchoptherinans have. So that's one on the right. I don't know that they've been studied very much, probably just because it's difficult to get close enough to an elephant to get a close look at its lice. And there's only a few species of those, but there is one of them diagrammed on the right. Sucking lice, certainly in humans, they can be transferred by close personal contact, sharing grooming supplies, clothes, bedding, things like that. Chewing lice can be spread among animals if they bed down together or share nests or something like that. Chewing lice spend their entire lives on a single host, and again, we call this being monoxenous. Lice can be transmitted from parent to offspring. So if a louse-infested bird is raising some chicks, some of them will probably jump onto the chicks. And lice may die when their host does, but they've been known to hitch a ride on flies. That's a blood-sucking hippoboscid fly, and on the left side of its abdomen, that little tan thing sticking off is a chewing louse that's hitching a ride. You might remember we used this word very early in the course. A relationship where one species gets transportation from another one but doesn't otherwise harm it is called foracy. So remoras hitching a ride on sharks would be foracy. There are human botflies and things like that may hitch a ride on blood-sucking insects. So may some nematodes, but they don't harm the blood-sucking insect. That would be another case of a phoretic relationship. There's a California condor. I believe that's the largest flying bird. And in the 70s, the species was almost completely wiped out, and the very last of them were captured for a captive breeding program. They have since returned to the wild, and they are making a slow comeback. But it's interesting that when the very last wild California condors were captured, they were deloused, dunked in some sort of medication that killed off all of the lice. And this wiped out Colpocephalum californici, a chewing louse that did not live anywhere but the California condor. Like I said, chewing lice can be extremely host-specific. And that means that as bird populations divide, chewing louse populations divide with them. We talked about this some time ago, but think of the model of speciation where you start with one population that gets split into two by some barrier. Anything that splits a population of hosts into two is going to split the population of parasites on the hosts into two. And that means the evolutionary history of parasites often matches the history of the hosts very closely. You do occasionally get host switching, but that may be more common in some groups than others. But very often in the chewing lice, you get very close matches between the evolution of the host and the evolution of the parasites. And in the case of Digiriella regalis, we're actually able to see that happening. So here's Digiriella regalis. I think that's a female on the left and a male on the right. And here's what it lives on. Buteo galapagensis, the Galapagos hawk. The Galapagos get talked a lot about in evolutionary biology because Charles Darwin made a famous visit there. You've probably heard of the Galapagos finches as a classic case study that provided evidence for the basic idea of evolution by natural selection. The Galapagos are little rocky volcanic islands 600 miles off the coast of Ecuador in the Pacific, right on the equator. And they're noteworthy for having species that are similar to what you'd find in South America, but not the same, and that are in fact unique to the Galapagos Islands. And in some cases, individual islands will have species that are unique to that island alone, like the Galapagos tortoises, for example. I mentioned the Galapagos finches, but there are something like 4 unique Galapagos mockingbirds. There's a Galapagos penguin. There are Galapagos rodents. There's a Galapagos tomato. There are Galapagos prickly pear cactuses. And there's a Galapagos hawk, Buteo galapagoensis. This is in the same genus as hawks you would see around here in North America. Red-tailed hawks and red-shouldered hawks come to mind. Buteo jamacensis and Buteo the other oneensis. There's also Swanson's hawk, which I don't know if we get much around here. You can ask Vicky if you want to know. And both the Galapagos hawk and its closest relative, Swainson's hawk, are parasitized by Digiriella regalis. Now, the Galapagos hawk lives on 8 of the Galapagos islands, the ones that are color-coded here. Isabela is the largest. That's the one in orange. Next to it, you've got the large Fernandina and Santiago and the tiny island of Pinzon. And then you have outer islands, Pinta, Marchena, Santa Fe, and Española. Hawks don't like flying over open water, so populations of these hawks don't mingle very much. Española hawks very rarely would fly to any of the other islands. There might be a bit more exchange between Isabela and Fernandina. Notice that those two are very close to each other. Santiago is also pretty close to Isabela, sorry. But certainly those outer islands are farther away, and hawks there are very rarely going to cross water to reach another island. What's been done is they took blood samples from a whole bunch of hawks, analyzed their mitochondrial genetic diversity, and were able to sort out unique alleles. What you're looking at here is basically a very tightly packed bar graph where each one of these bars represents the fraction of the mitochondrial genome of such and such a bird that consist of alleles that are unique to that particular population. So if you look on the right, you can see there's a band that's kind of maroon colored that says Marchena, yellow says Pinta, Santa Fe is kind of pinkish, and Española is labeled green. What that's saying is all of the birds that they sampled on Marchena, Pinta, Santa Fe, and Española had genotypes that were almost entirely consisted of alleles that were unique to those populations. What this is showing is that there's very little genetic exchange among the populations of Galapagos hawk on Marchena, Pinta, Santa Fe, and Española, those outer islands. Very rarely does a bird from any other island make it out there and bring in alleles that would be unique to the population on that other island. It happens very rarely. Okay, on the left, you can see there's bars for Fernandina, Isabela, Santiago, and Pinzon. Those also have distinctive alleles, but you can see there's a bit more genetic exchange between them. So some of the Isabela hawks have genotypes that are more typical of Fernandina, for example. That's why in that block labeled Isabela, you're seeing some blue bars kind of in the middle of that sea of red. Santiago, the distinctive genotypes are in red, but there's a lot of light blue bars in there. That tells you there's a lot of genetic interchange between Santiago and Pinzon. And there's a lot of genetic interchange between Fernandina and Isabela, none of which is surprising because those four islands are close together. And I guess it's more likely for hawks to disperse either deliberately or maybe by a storm or something like that. It's easier for hawks to disperse between those central islands. So that upper diagram, that's an analysis of mitochondrial genetic diversity of hawks. Below, that is the same study done on hawk lice, and it turns out that they're almost identical. Almost all of the lice on the hawks on Pinta have a unique genotype that is very rarely found in lice on the hawks on any other island. So all of the lice on Española, almost all of them have a genotype that is distinctive for Española, and very few have alleles that might suggest they could have originated on a different island. So what you're looking at is what you'd expect if speciation is happening. The different populations of hawk are not quite genetically different enough that we can think of them as separate species, but they're different enough that each one has distinctive genetic markers in its mitochondrial genome. And the exact same thing is true of the lice. Lice on one island are very rarely brought to another island because birds from one island very rarely visit another island. And the exceptions are on those central islands of Fernandina, Isabela, Santiago, and Pinzon. And as you can see on the map, they're close to each other. which means dispersal is more likely among those islands than it is among those far-flung distant ones. So the evolutionary history of the lice is in parallel with the evolutionary history of the bird populations, which is good to know. There was actually another study that's been done on Galapagos hawks. Unlike typical hawks, they're territorial. They live in these family groups, sometimes of one female and multiple males, which is kind of an unusual arrangement. And family groups will stake out a territory and they'll defend it. Non-territorial hawks have difficulty competing with this. A lone hawk will have a hard time defending a territory if his neighbors are a family group that cooperate and work together. So hawks that don't have a territory don't feed as well. They can't defend a large hunting territory. They tend to be in worse physical shape. On the y-axis, what you're looking at is, I'm not sure how this was calculated, but it's a measure of body condition, basically how healthy the hawk is. And the non-territorial hawks, the open circles, you can see on average are much less healthy than the territorial hawks. But what's more, they're also worse parasitized. The non-territorial hawks have more of the chewing louse called Copocephalum turbinatum, which I'm assuming is caused by the hawks that are less well fed and more stressed, having a less efficient immune system. Although it's possible it could go the other way around, that the more parasitized a hawk is, the weaker it gets and the less likely it will be able to defend a territory. So off the top of my head, I'm not sure of the direction of cause and effect. But this is neat that there is this correlation between parasite intensity, physical shape, and ability to defend a hunting territory. And there's lots of other instances of co-speciation. I can't remember where I got this, but this is a fairly recent study looking at the evolution of pelicaniform birds. That's pelicans. It's also boobies and frigate birds. These birds that may cruise over the ocean for, extended periods of time. That phylogeny is over there on the left, estimated by looking at molecules. On the right, that's the phylogeny estimated by looking at molecules of bird lice in the genus Pectinopegus. And with one exception, that little X that you can see above the center, that's the only case where the phylogeny of the parasites does not perfectly mirror the phylogeny of the birds. In every other case, every time the bird speciated, the parasites did too, and the phylogenies are practically mirror images of each other. So that's what you'd expect when you have extreme host specificity. So host specific that I'm told that sometimes you can work out evolutionary relationships of birds by looking only at the parasites that live on them. And that's what I've got for you about lice. I should have put it into a hip-hop song so we could have, lice, lice, baby, do-do-do-do-do-do, lice, lice, baby. Okay, I'm not going to get any lulls for that, so I will now proceed to stop recording.